Welcome everyone to this month's NextGen Precision Health Discovery Series. I'm really excited to introduce our speaker here in just a couple minutes. Before I do that, I do want to make a couple important announcements. First of all, we are using a system called Mentimeter, so menti.com. You can scan the code, the QR code there to get the link so you can submit your questions. Or if you're on Zoom, you can also submit those in the chat. Mentimeter is actually preferred because if you like a question, you can actually upvote it. And so it's really the best platform if you can use that. Second of all, I have a very exciting, very short announcement about a new program that's actually starting today, our Discovery Fellows postdoctoral program. So this is a system initiative where we're going to be starting a 2-year postdoctoral researcher support program. I think we have a slide there showing some of the details. And so the application is open, and that is going to close. The deadline for that is at mid-October, and the anticipated start date is January 2025. So the goal is really to emphasize cross-discipline collaborations. That could be biotech or academics, or that could be cross areas in academics. So please get the word out about this, because we want to really fund the best talent across the system. Last but not least, I really am excited to introduce our speaker, Dr. Michael Nichols. Dr. Nichols joined UMSL in 2004. He is the director of UMSL's Biochemistry and Biotechnology program. He is also the associate chair of UMSL's Chemistry and Biochemistry department. And Dr. Nichols also serves as the president of the American Society for Neurochemistry. Alzheimer's disease, as people are well aware, is a leading cause of death in Missouri and across the country. It's the 6th most common cause of death. And we've made some advancements in our understanding, but there are major gaps. I think there's probably not one of us in this room that has had somebody, a friend or a family, that's been impacted by Alzheimer's disease. I have a grandmother that has dementia, and I had a grandfather that passed away from Alzheimer's disease. So very motivated to find better answers. And so it is my privilege to welcome Dr. Nichols to talk about his work trying to advance therapies for Alzheimer's disease.
Thank you very much. Thank you very much, Dave. It's a real pleasure to have an opportunity to speak in this NextGen Discovery Seminar Series and to showcase some of the research going on in the Nichols Laboratory, but also to kind of share some of the latest progress in Alzheimer's disease, research-based, but also therapeutic-based for everyone. So one of the things I'm excited about is I want to harken back to a time about 6 years ago, actually, when there was a precision medicine summit that was at University of Missouri, Columbia. George Gokul, Rob Paul, and I went down and gave short talks, and the idea was to help kick off this precision health initiative for the system. Chris Spelling, Vice Provost at the time for research, now Vice Chancellor for research and development, came to my house, picked me up, and we drove down to Columbia, spent the day. Okay, so it was a really good exercise, but the ideas were just formulated at the time. And that brings us to today, where we have this NextGen Precision Health building, which is now going to be a center of excellence for the whole system. Very pleased that a lot of this has been expanded to all 4 campuses. So that can not only be a resource, but a place where we can share ideas and collaborate within the system. So very exciting that it's made it this far with some tremendous effort by many people, I'm sure. So I know we've got a diverse audience here and online. So at times it's going to be too much science, and at times it may not be enough science for your liking. It just depends on who you are. But I'm going to try to cover everything that I can and bring everyone up to speed so that you kind of understand the basis of Alzheimer's disease. So this is my outline. I'll get to some of the research that we're doing about halfway through. So I think most of us, as was just mentioned in the introduction, have had some interaction, most likely a family member, but it could be a friend or a colleague that has suffered from Alzheimer's disease. Okay? It's the most common form of dementia, and there's ways to distinguish between Alzheimer's disease and other forms of dementia, which may be more readily treatable. Okay? But it involves personality changes, behavioral changes, and difficulty with normal life. Down at the level of the neurons, there's a substantial loss of neurons, and the brain shrinks dramatically, and ultimately the disease is fatal. But I will hope to show you today that some progress has been made on those in the therapeutic area. Okay? So I think I don't need to spend too much time on the statistics. Everyone is well aware of the impact financially and societal that Alzheimer's has. I think the thing that really jumps out at you is the expansion of Alzheimer's disease in the next 30 years. Part of that is due to the aging population, but also the third bullet point, the amount of unpaid hours for caregivers, family or friends, and the amount of money that is involved in that is tremendous. So I think everyone is impacted by Alzheimer's disease, whether you're an older or a younger person. Okay? Here's something that really stands out to me for research-wise. And a lot of this is taken from the Alzheimer's Association website. It's a wonderful resource. I would recommend anyone going to that and looking up plenty of information. And all of that information comes from scientists and clinicians on there. So we've made a huge amount of progress in many diseases. But the one that stands out is Alzheimer's disease. And I lump that in with other neurodegenerative diseases like Parkinson's or ALS, and there's others as well. Okay? So this is really the challenge for the next 20 or 30 years, and it has been a challenge. And so, yes, we can translate that progress in these other diseases and hopefully make that same kind of impact with AD. Okay? So let's get to the pathology, and I always start with the two main proteins of Alzheimer's disease. So Alzheimer's has been called a plaques and tangles disease. And what you see is a neuron with lots of axons reaching out, and inside that neuron is an accumulation of these tangles that are formed in the Alzheimer's brain. Outside of the neuron, and you can see these large circular things, are these plaques that form. Okay, so this is a hallmark of Alzheimer's disease, the plaques and the tangles. Okay, so I'm going to spend a little bit of time telling you about the plaques and the tangles and what they are. And if you don't have Alzheimer's disease, you don't have these plaques and tangles for the most part. Okay. All right, so let's focus on the plaques. And the main component of these plaques is the amyloid beta protein, okay? Also called A beta, so I'll call it A beta probably from now on, just for short. Others call it beta amyloid, so if you see any of those anywhere on the news, they're talking about amyloid beta and these plaques that accumulate. So the red circles are the plaques in a section of brain tissue from an Alzheimer's patient. They're outside the neurons, as I said. If we focus in, and this is an electron microscope image, on these plaques in the brain, you'll see that the A beta has formed these fiber-like structures. Okay? So there's just thatches of these fibers there. And if you look at the bottom EM image, you'll see that rigid fibril structure. One nice thing is we can recapitulate these fiber structures and other types of intermediates in the laboratory. Okay? What you see in this next image, the third one there with the green and blue, the blue is the amyloid plaque, so the A beta plaque. Right around that plaque, the green is neurons, and they're damaged all around the plaque. Okay, so they're in contact or around that plaque, and when they are, they're damaged. Okay. Another thing I want to highlight is inflammation in the brain. And so our research is focused on that aspect of AD, but also some of the biophysics of the disease and the protein. But what you see on the far right image is microglial cells, which are the immune cells of the brain. So they kind of surveil the brain and try to get rid of things that shouldn't be there, okay, and respond to any areas of injury. The blue, again, is the A beta plaques. And the microglia selectively cluster around those plaques. Okay? And when the microglia are activated, they release lots of inflammatory molecules, and that creates this chronic inflammatory environment. So if you're talking about years of amyloid building up, you're talking about years of inflammation also playing a role. All right, so where does A beta come from? Well, we all make A beta every day. So in the green there is the A beta sequence. And what you see for those who aren't familiar with this is these are amino acid residues as part of a larger protein. So this is the amyloid precursor protein. We all have this protein. We all have the enzymes that end up cutting, and you see those in the square boxes, the beta and gamma secretase. We don't know the true function of a lot of these proteins and enzymes, but we have them in most of our tissues and cells. The enzymes work every day. We produce A beta every day, and that A beta gets cleared every day. But as you age, something changes, not with everybody, but with some people. And you produce or have the inability to clear the A beta. Okay. There's two main forms that are produced, A beta 40 and A beta 42. Okay, and that's the number of amino acids long that segment is. Okay. A beta 42 is the one that's more highly implicated in Alzheimer's disease. Just those two amino acids means that it aggregates into these fiber structures much more easily. Okay. There's other things on this slide, and I don't want to spend too much time on it, but there's mutations in this sequence of APP. And so if you have those mutations, you get early onset Alzheimer's disease, typically in your 40s or 50s. Later onset AD is a little different. It's sometimes called sporadic AD, and that happens typically after 65 years old, and the incidences increase as you age. But all of those mutations are related to the amyloid beta protein and this APP. Okay. All right, so what about tau? Same brain section slice, but now the circles are around the neurons, and the tau is inside the neurons. Tau also forms these filament-like structures. So aggregation of these proteins is a key event in Alzheimer's disease. So that term, aggregation, means one protein gathers together and clumps and forms these larger proteins. And it's not just a messy clump, it actually has this fiber-like structure. Same with the tau. So what you see on the right side is in the normal neurons, you have these nice straight axons that can transport proteins back and forth, which is important for our communication. Okay. Over on the far right in the little circle in the upper panel, that's healthy tau that's helping stabilize these microtubules and these axons in the upper right. In AD, In the bottom two panels, you see that you lose some of these axons. There's gaps there. The circles, the oval circles on the left side. And in the right side on the bottom is aggregated tau in this filament structure. So it's misfolded. It's doing something that it's not normally supposed to do, and then it loses its ability to help stabilize these axons. So there's two proteins that play a key role in Alzheimer's disease. Okay. So what's the relationship between A beta and tau, the plaques and the tangles? Well, let's focus on this slide. And at the bottom, you see the preclinical AD, and then the symptomatic AD. So a lot is happening before you even have symptoms. And that's one of the keys to therapeutics and research. Okay. In the red line, I don't know if I have a pointer here, but the red line is showing the A beta deposition, or the plaques. Okay. So that is the real trigger for Alzheimer's disease. Along with that, the green line is the activation of inflammation with these microglia and other brain glial cells. A little bit downstream and time-wise is the tau tangle formation. Okay. But once the tau tangles start forming, the death of the neurons really starts to progress. So there's lots of questions here. Tau is most clearly associated with the clinical parts of AD, but without the A beta, you don't get the tau tangles in Alzheimer's disease. Okay. So that brings up lots of research questions. I can't touch on them all today. In fact, I better stay on time here so that we can have a question and answer period. But what's going on before the plaques are even deposited? Okay. Lots of things, I'm sure, and that's the key to really kind of intervening with Alzheimer's disease. What's happening between the plaques and the tau tangles? We've not worked that out yet. That's an important part, because if you can stop that part, you don't get the progressive decline in the neurons, or maybe you don't. And then how are the tau tangles killing the neurons? Big question there. So lots of questions, lots of people working on these problems everywhere, across the country and the rest of the world. Okay. So again, I want to focus on aggregation. It's a big part of our research, but it's a big part of Alzheimer's disease pathology. Again, when the proteins are in their normal native state, they're fine. Okay. But when they begin to aggregate into these bigger structures, that's when you start to have problems with Alzheimer's disease. So this is kind of a simple form of A beta aggregation. And we focus on A beta, so I'll spend my time on that. There's lots of things that you can do to change this aggregation process. Okay? You see those listed at the top. Fortunately, you can recapitulate a lot of things in the lab. Not everything, but a lot of that is very similar to what could be happening in the brain. A little more complex figure shows lots of other oligomeric states of A beta before it gets to the final plaques at the bottom right there. Again, the protofibrils are an intermediate structure. Okay, and then these oligomers, some are on pathway and some are off pathway for that. Well, you can follow this in the laboratory. There's lots of techniques for that. So the aggregation of A beta into these fiber-like structures, or fibrils, we can monitor in the lab with a molecule called thioflavin T. Okay, and that molecule exhibits fluorescence. It only binds to the aggregated material. It will bind to the protofibrils and the fibrils. It won't bind to the monomers. And so you can visualize that with fluorescence in the laboratory. That thioflavin T molecule becomes important on the next slide. Okay? So what do we know about these aggregation species? So protofibrils and fibrils have this beta sheet structure, which is very well known for protein biochemists, may not be as known for other people, but it's a particular way of folding, and you can detect that type of structure. So only the later stages have that structure. The fibrils themselves are actually insoluble. So you can see those in solution, and they deposit in solution. The protofibrils are soluble, and some of the other precursors are as well. We don't know much about the oligomers. The protofibrils are a little bit more well known in their structure. Okay. All right, so where are we with diagnostics and therapeutics? And the reason I wanted to get into some of the science with A beta and tau is because it becomes important when you think about the diagnostics and the therapeutics. Okay, so I kind of wanted to bring everyone up to the same level as well as I could. So a big breakthrough was made around 2005 to 2010. And that was that we are now able to image patients as they're going through the disease, or even before that. The compound, Pittsburgh compound B, or PIB, is based on that thioflavin T molecule that I just showed. But they made a radioisotope out of it, and you can use it, a PET scan, to image the amyloid. So ever since then, now we've had a way of detecting the amyloid building up in the brain. Okay? We haven't really had a way to stop it. But that's a key thing, because if you can detect it and look at it and you start to make progress on stopping it, you can see that, okay? And you can assess things. Okay? So you see a normal brain on the left, and then the AD brain has lots of amyloid in it, and that's that PIB compound. Okay. So one of the key diagnostics now is, unfortunately, it's cerebrospinal fluid, which involves a lumbar puncture. So it's quite invasive. Not everyone's crazy about going in and getting lumbar punctures all the time. But it's a combination of phosphorylated tau, total tau, and A beta 42. So I've introduced you to those proteins, and that's one of the diagnostics in cerebrospinal fluid. It has a pretty good accuracy for AD, and it can differentiate AD from other dementias, which again is important to do so. So a very recent and very promising finding is, and everyone's been searching for a blood test for Alzheimer's disease. I think we would all agree that's better than a cerebrospinal fluid test. So this is 2024, Journal of American Medical Association, and a company that's here in St. Louis, C2N, is at the forefront of this with some WashU scientists that run that company. And that's a combination of phospho-tau 217 and a ratio between A beta 42 and 40. I think you'll find this interesting. So if you just go to, they evaluated, I think, 800 or 900 patients, went to their primary care doctor, and they were about 61% effective at diagnosing the AD, called in a specialist, about 73%. The blood test was 91% accurate. So after these patients saw their doctors, they then went to more confirmatory tests that we have available to show whether they had AD or not. So that looks very promising, and that's really important. And the key is, how early can you test these, and how early do they show some indication that you might get Alzheimer's disease? Because if you remember that time graph that I showed, you want to try to get to things before all of that starts happening, the plaques and the tangles. All right, so what about therapeutics? Up until about 3 years ago, everything was a non-disease modifying therapeutic. Okay, so these are things that kind of address the symptoms. They'll maintain acetylcholine levels. They don't really stop or slow the disease. Or they'll, block dysregulation of calcium. And they're good medicines. Okay, they can buy some time, but they don't directly address the disease itself. And about 3 years ago, FDA started approving some antibody-based therapeutics. Okay, and that's on the right side of the screen. Biogen has 2 of those. All 3 of these are anti-A beta antibodies. And the top 2 are selective for aggregated A beta, so protofibrils and fibrils. The bottom one is selective for another type of aggregated A beta, so it has a modification in it. Biogen is discontinuing Aduhelm at the top because lecanemab is being administered to patients in the clinic. So these have been through all the clinical trials, and we'll see optimistically how they work as they're being administered to patients. Kisunla has just been recently approved, so that hasn't really been rolled out yet. Okay, but again, they're all antibody-based, so you have an IV infusion of these antibodies. Okay, so the latter 2 treatments there were shown to modestly slow cognitive decline. Okay, so I'll touch on it in my concluding remarks. We're not quite there yet, and it's obviously going to take a lot of other things, and it may require multiple types of medications instead of just finding one particular target. But this is all good, promising information that we're starting to make some headway. And believe me, everyone is learning from everyone else. Okay, so a little bit about research in my lab. See how I'm doing on time, okay. So we are working in several areas. We're always looking at biophysical mechanisms of A beta aggregation. I think it's really important to understand what's going on with the aggregation so that you can apply it to biological environments. So with that being said, we focused on inflammation. So I tried to highlight that in the slides, that inflammation is an integral component of Alzheimer's disease and other diseases as well. Okay, so these are our 3 areas. I'm going to weave through inflammation all the way through the rest of the talk. Okay, but I am going to sprinkle in some biophysical work. So this is where it could get a little bit hairy on some of the science, and I know we have lots of different disciplines, so even scientists of different disciplines don't always completely understand what each of us are doing. That's why interdisciplinary research is good to share that knowledge. Okay, but those latter 2, when I talk about inflammation, we're really focused on the immune cells. So microglia, peripheral monocytes we use, and macrophages as model systems. But that's kind of a rough image of a microglial cell or an immune cell. All of these things, you'll see a little bit of it sprinkled through in the next 20 minutes or so. Okay, but these are all areas that we're working on. When you think about the architecture of a cell, the next slide and why we do biophysical studies kind of tells you, the reasons why. Okay, it's all about interactions. How do things interact? Proteins and cells, proteins with each other, other types of interactions. All right, so I've got one slide with kind of a lot of biophysical stuff. I know one of my colleagues will appreciate some of this here. So why are we interested in trying to understand these distinct species? And it has to do with the size, the structure, the types of exposed surfaces they have, and also their internal environment. So how do you get in there and stop it? How does it interact with a receptor on the outside of a cell? Lots of other types of interactions. So one of the things we did a while ago was make these tryptophan substitutions in the A-beta sequence. Okay, so that's the A-beta sequence. We replace phenylalanines with tryptophans, but only one at a time. So you see the numbers up there at position 4, 19, and 20. We had 3 separate A-beta peptides with these different tryptophan substitutions. On the right is a structure of A-beta that was determined, the A-beta fibrils, by solid-state NMR, by Rob Tico's lab. And you can see that the red circle is the 19 position pointing in, and the blue circle is the 20 pointing out. Okay? Well, the good thing about tryptophan is it fluoresces, so it displays fluorescence, and most fluorophores, depending on where their fluorescence shifts to, can tell you about the environment that it's in. Okay? And so what we see here, all of these peptides, proteins form these fibril structures, which I'll show in a minute. But only the F19W, that's the tryptophan fibrils, shifted to the left. So what does that mean when it shifts to the left? That means it's in a greasy or hydrophobic environment. So it tells us what the environment is like for that amino acid. When we compared the F19 and the F20 tryptophans mutants or substitutions, comparatively, only the F19W one shifted again. So that one is in a greasy environment, okay, while the other ones were not. And that kind of matched that structure that we see above those graphs. They all form these fibril structures. Again, these are electron micrographs of the fibril structure. So there was nothing different about them, but they served as a probe for that structure. So we took it a little bit further, and we wanted to look at not just the fibril species, but the soluble species. So in that upper right-hand corner, but in the graph that shows the peaks, that's a size exclusion chromatogram. That separates things by size. The biggest things come out first. So all those little peaks, they're getting smaller in size. But they have different surfaces. So if you look at the graph on the right, the ones that are further to the right in the chromatogram bind to this molecule called bis ANS, which means it has exposed hydrophobic regions or greasy regions that are outside. Okay? If you get to the protofibrils, they bind the THT in the upper right-hand corner, but they don't have that exposed greasy area or hydrophobic area. So what did we learn from that? That these tryptophan substitutions can be very valuable as a probe. But there's lots of other smaller, less defined structures. They're called oligomers that have more exposed surfaces. They're not as tightly folded. They didn't insert the tryptophan as well. But the more highly structured soluble A-beta species are the ones that induce the inflammatory response the best. And that you'll see in just a minute here. Okay. So when we first started, we wanted to know which species is the best inflammatory one for A-beta. And what you see on the left-hand y-axis is a molecule called TNF-alpha. So that's an inflammatory molecule. Its levels are raised in Alzheimer's disease. It's an important inflammatory molecule in other, even arthritis and other types of inflammatory diseases. But it's important in AD, and so what we did is we allowed the A-beta 42 to aggregate over time. And we kept testing the cells to see when the best response would happen. And we saw it right in an intermediate time frame. Okay? We did lots of studies to try to characterize what that time frame was. Okay? We found that it was very soluble. So even at 100,000 times G, we couldn't spin that species down. We used an antibody called OC that recognizes elements of fibril structure, but even across soluble species. And you can see in the third bar on that graph on the right that it completely immunodepleted that species, and so we no longer got a response. Okay. We ended up purifying that species. We did an atomic force microscope analysis, and this is what we saw. So this is very reminiscent of what other people had seen before us of protofibril structures. And that led us to really focus on protofibrils. So we have ways to prepare protofibrils in the lab. We can use that size exclusion chromatography to separate protofibrils from monomers. We can also prepare fibrils in the lab, as you've seen. So we can do comparisons here. So we now prepare our protofibrils in an artificial cerebrospinal fluid, okay, to try to get it to physiological conditions. There's also ways that some may not know this technique. It's called circular dichroism. The red line is very indicative of a beta sheet structure for proteins. Protofibrils have that structure, the monomers don't. Okay. If we look at them by EM again, you see the protofibrils are much smaller than fibrils. Fibrils are long branch structures, protofibrils are soluble, curvilinear, much, much shorter. Okay. We also do a lot of biophysical light scattering techniques. Sounds complicated, it is kind of complicated, but essentially it measures the size of things, molecular weight. Okay. So we know that protofibrils are much bigger than a protein like BSA, but they're still very soluble and diffusible. And we used another technique called multi-angle light scattering to determine the molecular weight of these protofibrils. So they have about 200 to 2,500 monomers in them. So they're pretty good sized. Okay. All right. So here's where we get to the cell work. Okay. So we looked at microglial cells, wanted to see what kind of forms of A beta would bind to the cell. So this is confocal microscopy, and it measures fluorescence. So you can use antibodies that have fluorescent tags on them to find your protein or species that you're trying to look at. Okay. These really show up better when it's dark, but it's fine here. So what you can see if you go from left to the upper panel, left to right, and then bottom panel, left to right, that only the protofibrils really bound to the microglial cells on the surface. Okay. So the A beta protofibrils are in the green. All right, so if we look at that thioflavin T fluorescence again, that I've mentioned a couple times, we can see that fibrils have much more. This is all based on the exact same concentration of each one of these A beta species. Binds to thioflavin T, shows much more fluorescence, much more than the protofibrils, but the monomers don't have any. Okay. So the protofibrils are a precursor to the fibrils. They haven't fully developed that fibril structure. But when we treat these microglia or immune cells, again, and we measure that TNF-alpha protein, we see that the protofibrils induce much more inflammation than the fibrils or the monomers. Okay. So they're quite active. In a study following that, we fluorescently labeled A beta protofibrils and monomers, and we did this confocal fluorescence study again, and we looked at the microglia and how well they could take up the A beta, get inside the cell. So these are primary neurons taken, primary microglia, taken from a mouse model, and then we did the experiments on these cells. And I think by eye you can tell that the protofibrils get into the cells much more easily than the monomers do. Okay. And these are just snippets from different studies, and the right-hand graph is just kind of a quantitation of the confocal. We also did the study a little bit differently in a dish with different wells, and we added the A beta protofibrils and monomers to the cells, and then we washed those cells, and then we probed the cells for A beta getting inside the cell, and it matched what we saw with the confocal. We took it out a little bit longer, so you can tell these are just short time frames for the confocal. But with the dish-based assay, we went out to 24 hours, the A beta is still inside the cell. It's not being degraded inside the cell. Okay. Monomers, again, didn't get in very well in the lower part of that right-hand graph. So just to summarize some things at this point, I've told you about the protofibrils. We've done other studies. A beta 42 is a really key part of any mixture of A beta 42 and 40. It's extremely stable once it aggregates. We've also shown that the A beta down at the second to last bullet point can get inside the cell and then be packaged into these vesicles that are then released from the cell in the microglia, and that's a whole nother study. And so that has implications as well. So I know I keep mentioning the plaques, but I want to digress just a little bit because I keep saying the plaques are full of fibrils. But if you look at this study by Brad Hyman's lab, what they found in the middle panel with this confocal fluorescence, they used an antibody that was selective for oligomeric A beta, and it hovered around the plaque. So the blue in the left is the fibril part of the plaque. Around the plaque is this oligomeric or protofibrillar A beta. So there's a lot going on around these plaques, and it may explain why the microglia are clustering around these plaques and in a highly activated state. So nobody's really returned to this, but I find it very interesting that there's a lot we don't know about the plaque environment there. So because of all this data we kept seeing with the protofibrils, we decided years back to try to develop a protofibril selective antibody. So in collaboration with Pacific Immunology in California, and when I say collaboration, we mailed stuff back and forth and talked all the time, they immunized rabbits with our purified protofibrils. And we gave them, they sent us the serum back, and in fact that serum in the right-hand graph was selective for protofibrils over other forms of A beta. Okay. So we followed that up. These are different ways to show this selectivity. On the left, you again can see the selectivity for the protofibrils, and that's by adding these different species in the bottom of a well and then adding our antibody on top of that and see which one does it recognize, the antibody recognize the best. If we take that concentration range out, you can clearly see that it shifts to the right. That means it has a much weaker affinity for that type of A beta, our antibody there. So on the left is the protofibrils in blue in that right-hand graph. Okay? In collaboration with Colin Combs' lab up at University of North Dakota, we used some mice that had been engineered to get Alzheimer's disease. This is a whole other topic that I probably can't spend much time on. They have the human form of the amyloid precursor protein in it, and they have the enzymes, so the mice can generate the plaques if they have the human form of A beta. Okay, now there's some limitations to mouse models, but they do generate the plaques. And on the left is 4G8. That's a conventional A beta antibody there. In the middle is our antibody. You see some overlap in some places, and in other areas you see no overlap at all. So our antibody is recognizing different elements of these plaques. On the right side, the upper two sets of panels are the transgenic mice that have the plaques, and our antibodies in the green, the conventional antibody is the 4G8. It's very sensitive to this A beta accumulation. The bottom panel is just a regular wild type mouse. Doesn't generate the plaques, and you don't see those there. So this next image kind of shows at higher resolution the difference between our antibody in the green and the conventional A beta antibody in red there. Okay. So we pushed further. We worked with Pacific Immunology and Exxon Bio to develop a monoclonal antibody, which is kind of the gold standard in research and therapeutics for that matter. And in fact, over time, it took us a lot, and the companies helped out, so did my colleagues here at UMSL, and lots of conversations. We were able to clone these antibodies, develop them, sequence them, express them, and purify them here on campus. And you see a variety of assays. When there's a little M there, that means it's the monoclonal form of the antibody, not the serum antibody. Some are very selective for protofibrils. Others are very good for using as a control because they're not selective, but they were generated in the same way. Okay. So in a little bit more rigorous way of determining the affinity of these antibodies, this is a competition ELISA, and you can see in the left panel the protofibrils versus the monomers. If it's shifted to the left, it means it recognizes much better, has a higher affinity. So you can see the monomers comparative and the fibrils and the protofibrils in comparison. Why is that important? So what does selectivity mean? So if we think about the antibodies that have been approved, aducanumab and lecanemab, they recognize fibrils and protofibrils with the same affinity. Okay? And so you see that down in the lower panels. If you look at oligomers or protofibrils, you see the same spot. So those are dot blots and are using the antibodies there. They don't differentiate between those two, but our antibody does, okay, between the fibrils and the protofibrils. So that may have implications for how much of your antibody gets sucked up by different species of A beta in the brain. Okay. So we've done lots of other studies with the antibodies, and we're trying to figure out even new ways to research that and make those available for other people to use as well. All right, so I'm going to run out of time, but I want to go through some recent data. So when I say that A beta protofibrils interact with the cell, what do I mean? So there's a family of receptors called the toll-like receptors. They typically recognize bacterial and fungal components. But we found that A beta 42 protofibrils interacted with those receptors. Okay? It initiates a pathway that generates TNF alpha. And we found that if we block those receptors, and there's multiple receptors, we could diminish the response, and that's the middle bar there. Okay. We used a different technique. So we used mice that had MyD88 knocked out. So that's the protein in red. Okay. And when we did that, we see we get a concentration or time-dependent production of TNF alpha. But if the MyD88 is missing, it blocks the response from A beta. Okay. And quite a bit of the response. So we feel like the toll-like receptors are generating a lot of this pathway. Okay. Now, a second cytokine that gets produced is this pro-IL-1 beta. In order to get mature IL-1 beta, it takes this inflammasome complex of 3 proteins to get activated and produce the mature IL-1 beta, which can get secreted from the cell. So that typically takes a second signal for that to happen. Okay. And what we found is that, again, mature IL-1 beta was being produced inside the cell, and that could be blocked with the MyD88 knockout mice. Okay. And so we also found that mature IL-1 beta was getting secreted from the cell. Okay. So what that told us is that A beta could do both signals. And others have now confirmed that. And that tells you that you have a protein inside your own body, not a bacteria or fungus, that can activate these inflammatory pathways inside yourself. So we're very interested in this inflammasome complex. So we started a project to try to study those 3 proteins. You see the 3 proteins on the right, NLRP3, ASC, and caspase-1. Okay. So what we've done is recombinantly express these proteins in the laboratory so that we can do lots of studies with these proteins. Okay. So we purified these by chromatography again. On the lower right-hand side is a Western blot to show that we can express and purify these proteins. These proteins are also very difficult to work with. They oligomerize quite easily. And you can see that on the left panel, you get oligomers of this ASC protein. You can see in the middle panel that we can purify the native protein on the right, so you only get 1 band. Or if you look at the aggregated material, you can see that there's lots of bands. And the red line shows that it has a native alpha helical structure. So we have these proteins in the lab to study, but it also leads into our most current project that we're working on now. And that is, how does A beta stimulate the inflammasome? Okay, so the most well-established mechanism is an indirect way. By A beta getting into the lysosome breaking apart, and then an enzyme being released that activates the inflammasome. The hypothesis we're following is that there's a direct activation of this complex. Okay, and there is some indication for this type of direct activation. Just a couple of years ago with some other type of protein that forms fibrils itself. Okay. So we commence studies. We know that these inflammasome proteins in our hands can inhibit the aggregation of A beta. So in all of these slides, you see A beta 42 by itself. But if we keep increasing the amount of protein, ASC, NLRP3, then they inhibit the aggregation of A beta monomers. Okay. On the right panel, we had a subunit that we expressed, and it did not inhibit the aggregation. We took advantage of our proteins having a certain tag on it that we could pull down. And we incubated A-beta together, A-beta are from these aggregation solutions with the ASC and the NLRP3. We pulled them down with the beads, and then we probed for the A-beta, and only the proteins, NLRP3, ASC, have the tag on them. And in fact, when we eluted that, they had the A-beta attached. Okay. Another scientist in the lab, Nasha Makoni, had some early data showing if we treated cells with the A-beta, it got into the cells, and if we pulled down this NLRP3 protein, on the very right side of that, it pulled down A-beta with it. Okay. So what are we doing now? Cristina Sanobis Pereira is here, and a lot of this is her data on the next two slides. We're trying to construct a model in the HEK293 T cells, which are not immune cells, but they're great for transfecting proteins in, and they don't have a lot of the endogenous proteins already in there. So you can basically build your own protein machinery inside the cell. So Christina has taken those plasmids that we already had from the previous project and in fact transfected the 293 T cells with each one of the proteins. So each one of those have a non-transfected and a transfected protein, and below is the alpha tubulin. Since it's inside the cell, that's a control protein that should be there whether it's transfected or not, and it is. Okay. She also has shown that the A-beta can get inside the cell with these HEK cells, and they're not really an immune cell, but the A-beta can get inside there. So on the left in the middle, at that last little dot, means that the A-beta has gotten inside the cell after lots of washes and then splitting open the cell. Okay. So she's also quantitated that with an ELISA. And again, we can see that the protofibrils get inside the cell much better. Okay. So she's just moved to confocal microscopy, and you can see that all three proteins now have been expressed in these cells. Okay. So lastly, what we're doing is now trying to do those pull-down assays in these cells. Okay. And so we use the beads again that recognize this tag on the proteins. The A-beta has gotten into the cell. Okay. Christina has washed that and then eluted the beads, and again they have the A-beta attached to the ASC and the NLRP3. And just in the last week or so, she's transfected with ASC, treated with A-beta protofibrils, and in yellow is where there's co-localization between the A-beta in green and the ASC in red. So we're seeing some yellow co-localization, lots of work to do, but that's what we're working on right now. Okay. So just some concluding thoughts here. As I mentioned before, it's been slow, therapeutic-wise for AD, but lately there's been steadily advancing progress with AD treatments. A-beta and tau aggregation has lots of different species, but it may be very beneficial if you can target a particular species or not. I think I've already summarized most of that, but the point of most of our studies and others across the country is that if you try to understand the pathways by which AD is being mediated or progressing, it provides ways to try to stop that or block it therapeutically. That's where all this basic research kind of comes into play. Okay. Lots of support for these projects over the years, department-wise, university-wise, system funding, external funding as well. And then, of course, lots of people that have helped at all levels, Ph.D., masters, lots of undergraduate researchers that have gone on to do great things. So with that, I went a little longer than I wanted to, but thank you very much for your time.
So we've got a lot of questions here on Mentimeter, a lot of engagement, but because we're low on time, I'm just going to pick the top one. A lot of people want to know, considering the known inflammatory response with microglial activation, have you considered anti-inflammatory treatment methods to slow down AD progression?
It's been discussed and debated for many years. When I was a postdoc at Mayo Clinic, we talked about this quite a bit. And there was some indication that anti-inflammatories, including some anti-cholesterol drugs, might play a role. I don't think they seem to stop the progression of the disease. So they're more of a peripheral thing. But again, I think a lot of it has to do with the timing. And so a lot of the things that are occurring, I think if we can possibly intervene earlier, all of them may have a way. But certainly attacking inflammation while you're attacking some of the main components of the disease causation, I think is probably going to be a strategy.
So for the sake of time, unfortunately, we can't. There were lots of good questions on there, and hopefully that people can connect later and ask those. But beautiful talk. I would like to thank everybody here in St. Louis and also online that's joining us for this talk. And most of all, Dr. Nichols, thank you for your work that is impacting the field and trying to make some progress on patients' care with Alzheimer's. And as a token of our appreciation, we would like to give you this, which represents that effort to impact people here in Missouri and beyond. And so thank you so much for sharing your work and for all the excellent, beautiful work and the team that has contributed to this.
Yep.
Thank you.
Thank you. Thank you.
So I'd like to welcome everybody September 24th, our next Discovery Series. We have Yael Weiss, who is a physician scientist, CEO of Mazi Therapeutics. The goal of Mazi is really to accelerate discovery and care for patients with ultra-rare diseases. And so it should be a very engaging talk. There's a big summit in Kansas City right around the same time, the same week that Yael is going to be attending as well. So we're really excited to host her for that visit.